that we have studied in the previous class. So enzymes are protein that acts as biological catalysts as accelerating the chemical reactions. So antibodies are also proteins that protect you when unwanted substance enters into the body. Antibodies also acts as a proteins. What are enzyme inhibitors, Sandra? It is a substance that prevents enzymes from reacting with a substrate. So what are substrate then? Substrate andre it's nothing but the molecule or drug or a chemical substance. A substance is a molecule that if an enzyme react with means which reacts with the enzymes or with the protein. So in this unit 12, we are going to discuss with polymerization of DNA. So we are going to separate and identify the DNA, RNA, enzymes, and the protein structures. So what are the methods you are going to follow? So the the basic concept which you are going to use study in this unit are polymerization of DNA and nature of blood flow throughout the vessels and detection of intra disulfide bonds in the protein. So intra disulfide bonds, how it has been connected andre by the peptide linkages. So when the one amino acid is linked with another amino acid, there will be formation of ಹಿಂಗ್ ಕನೆಕ್ಟ್ ಆಗಿರುತ್ತೆ by bond ಅಲ್ಲ, so there will be formation of disulfide bonds also, there will be formation, so that we are going can identify detection of intra strand disulfide bonds in protein. Also one more method, gel electrophoresis. Electrophoresis andre it is the migration and suppression of charged particles under the influence of an electric field. So when the electric field is applied, it can migrate or separate the charged particles. So that we are going to study different types and also the sequencing, DNA sequencing and DNA fingerprinting, conformation of nucleic acid and protein charges. So protein andre it's nothing but the enzymes charges present inside that. It's also a chemical compound, Allah. So there'll be charges present. So how we are going to identify that protein charges that we are going to study in this unit. So first, basically, what is polymerization? The polymerization is a chemical process in which small molecules called monomers are joined together to form large molecules. Formation of large molecules ke ಏನಂತಾರೆ polymerization ಅಂತಾರೆ. So small molecules when linked with large quantity. So that is known as polymerization. So it is generally used to produce wide range of materials. Bulk polymer solution polymerization, suspension polymerization, emulsion polymerization ಅಂತ ಇದೆ different types and interfacial polymerization. So why you are going to use polymerization andre it is used to produce wide range of materials including plastic, rubber, fibers and adhesives. So that's why polymerization is made. So polymerization is a chemical process in which small molecules join together to form large molecules are called as polymerization. So here DNA polymerization is process. Polymerization and ಅರ್ಥ ಆಯ್ತಲ್ಲ. So small molecules join together to form large molecule that is known as polymerization. So here sequencing of DNA polymerization. DNA polymerization is a process in which new strands of DNA are synthesized. ಅಂದ್ರೆ large molecules of DNA will be synthesized. It occurs during DNA replication or transcription. So it occurs ಯಾವಾಗ synthesis you are going to make ಅಂದ್ರೆ it is both DNA replication or transcription can be done. The DNA replication of cell genetic material is duplicated in preparation of cell divisions. So this process of DNA polymerization involves the formation of phosphidine ester bonds between the nucleotides. So between the nucleotides, the phosphorine ester bonds are formed, which are building blocks of DNA. So when the linkage between the two nucleotides, the phosphorine ester bonds are formed. So such DNA polymerization process will be taking place. So coming to the point, next topic here, the polymerization of DNA and natural nature of blood flow through different vessels. So how the polymerization of DNA take place? Polymerization of DNA also known as DNA synthesis. So synthesis of DNA is nothing but the addition of small molecules to form the bigger or large molecule. So here polymerization of DNA andaga addition of nucleotides will be formed. So synthesis is a process in which the nucleotides are joined together. So what are nucleotides ಇಲ್ಲಿ? Nucleotides are nothing but the basic building blocks of nucleic acid. Nucleic acid ಅಲ್ಲಿ ಏನಿರುತ್ತೆ? DNA and RNA will be present inside the nucleic acid. So nucleotides ಅಂದ್ರೆ they are the basic building blocks of nucleic acid. So polymerization of DNA means polymerization is nothing but the process in which nucleotides are joined together to form DNA strands. So polymerization of DNA is nothing but the synthesis, the process by which nucleotides are joined together to form a DNA strand. Nucleotides andre it is the basic building blocks of nucleic acid. So the presence of DNA polymerization involves the formation of phosphodiester bonds between the three hydroxyl group of one nucleotide to another nucleotide. So one nucleotide molecule will be linked with another nucleotide with the formation of the bond. The phosphodiester bond will be formed. So this is what the DNA polymerization. Here DNA polymerization can be studied by how it can be studied on studied by viscosity study. When DNA polymerization occurs, the chain length of DNA increases, hence the viscosity also increases. DNA polymerization ಆದಾಗ the chain length, because one nucleotide in the ಇನ್ನೊಂದು nucleotide it will be adding, it will be going on synthesizing ಅಂದಾಗ it will be added ಅಲ್ಲ, one nucleotide to another to one more nucleotide will be added. So the polymerization will be taking place. That means the chain length will be increasing. Chain length of DNA increases, hence the viscosity also increases. So if there no change in the viscosity indicates absence of polymerization of DNA. Viscosity li ಏನು changes ಇಲ್ಲ ಅಂದಾಗ there is no polymerization will be taking place in the DNA. So for example, where they are given three types of DNA reaction taking place. See, first in the first one, circular DNA molecule plus nucleotide, no change in configuration equals to no change in viscosity. So circular DNA, it will be in the form of ಹಿಂಗಿರುತ್ತೆ. So this is in the form of circular DNA. There will be strands formation. So in the molecule nucleotide, there will be a single strand. So no change in the conformation, hence no change in the viscosity. When circular DNA molecule plus nucleotide in the double strand helix, the linear DNA increases and the viscosity is observed. So viscosity also increases in the second step, and the circular DNA molecule plus again nucleotide, there will be multiple strand formation. So multiple small fragments will be formed and large viscosity can be seen. So before knowing about DNA, it is made up of uh, adenine, thymine, guanine, and cytosine. These are amino acids. Of these chain links, the formation of DNA it can be seen. So these four amino acids, the linkage is known for DNA. ಯಾವ್ದು ಅಂದ್ರೆ adenine, thymine, guanine, cytosine. So what are the role of nucleotides andaga? Nucleotides are nothing but it are the basic building blocks. So these nucleotides you should be knowing what are the roles that plays inside the body andaga. The building blocks of nucleic acid and storage of chemical energy. Also, the carriers of activated metabolites for biosynthesis and metabolic regulators. It acts as carrier inside the metabolic synthesis. Synthesis of biosynthesis ಆಗ್ಬೇಕಾದ್ರೆ it acts as a carrier, and also building block of nucleic acid and metabolic regulators. So when the viscosity is, when there is no charge change in viscosity, that indicates absence of polymerization. When there is a change in the viscosity, there is a presence of polymerization of DNA can be identified. So this is the method we are going to identify. How we are going to identify the DNA polymerization is taking place or not, and that it is depending on the viscosity of that the DNA polymerization is taking place or not, it can be identified. So the viscosity once again decreases and the viscosity of circular DNA increase and decreases in figure. So here you can see A is represented as circular DNA and B is represented as linear DNA and C, D are represented as multiple fragment of linear DNA. So should be knowing what is circular DNA, linear DNA. So this is what the circular DNA. So linear DNA ಅಂದ್ರೆ it will be like this. So the structure of linear DNA, and this is the structure of circular DNA. So when it comes to different types of polymerization, here the circular DNA, it will be in the linear, and when it comes to linear DNA, it will decreases when once increases and then it decreases. Therefore, the viscosity explains the polymerization of DNA. So here with respect to time, the which is taken in the x-axis and nu which is the viscosity. So how the viscosity with respect to time. So circular DNA, it will be always stable and linear, and when it comes to the linear DNA, it increases the viscosity of the linear DNA, that is B, will increases the viscosity of linear DNA will increases and then decreases with respect to time. So we can study scene explains the extent of polymerization in DNA molecule. So one more plot they are given here, that is erythrocyte versus hemocyte. So hemocyte ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ? Erythrocyte ಅಂದ್ರೆ erythrocyte is nothing but the red blood cells. Red blood cells na it is denoted as erythrocyte. Also, which contains hemoglobin. That is known as red blood cells. Is known as erythrocytes. And hematocytes andre it's nothing but the property of red blood cells in the blood. So here we are going to measure the diameter of those vessels of the curve flattened between the large vessels. So here it represents the plot with erythrocyte versus hemocyte place a different diameters in vessels shown below. So different diameters the present. For smaller diameter, the vessel curve is flattened, but for larger vessels, it is high. So the blood vessels in bioconcave and flexible membrane becomes of property cells. The apparent viscosity of man is termed hematocyte. So the viscosity of man in terms of hematocyte vessels with charge of shearing stress. So the value will be around 45%. So variation of viscosity of blood influences the working of art. So how thinner or thicker the blood, the viscosity of that it depends. So generally the man ಎಷ್ಟಿರುತ್ತೆ viscosity it will be of hematocyte the value will be around 45%. So nature of blood flow through different vessels. So depending on the vessels, the blood flow may increase or decreases. So blood flow through different vessels in the blood varies based on their size and the structure of the vessels. The blood flow is faster in larger vessels such as aorta and slow and also slowest and small vessels such as capillaries. Capillaries are smaller vessels, so the blood flow will be very slow, and aorta are larger vessels where the blood flow will be high. So this is the nature of blood flow through different vessels. Depending on the size, the blood flow will be taking place, and the presence of walls in the veins helps to prevent the walls present in the veins which prevent the reverse flow or back flow and maintain the flow of blood in the single direction. So another important point, the presence of walls in the veins helps to prevent backflow and maintain the flow of blood in the single direction. So, what are the effects of viscosity on nature of blood flow through different vessels? Viscosity means how it affects the blood flow through the different vessels. Why this topic ಏನಕ್ಕೆ ಬಂದಿದೆ ಅಂದ್ರೆ the viscosity because of DNA polymerization. So it depends on the polymerization of DNA. The viscosity may increase or decreases. So depending on that, the blood flow will change. So here how the viscosity of nature of blood flow through different vessels. On different vessels, the flow will be of different. So viscosity plays very important role here in the circulation of blood in the human body. So the blood is high viscosity, highly viscous, and is affected by even a small change in the viscosity of the medium. The blood flow varies inversely with the viscosity of the blood. So it mainly depends on the flow of blood. If the viscosity is high, the flow will be very less, and the if the viscosity is less, the flow will be high. So depending on the veins also, so depending on the viscosity, the blood flow will vary. So the percentage of blood occupied in the erythrocyte, one of the major component of the blood. So such as for larger vessels like aorta and veins, hematocyte value is high. So means high andre the surface area of the vessels will be high. So for capillaries and small veins, these are different veins in Lidela. So larger veins such as aorta and veins, hematocyte value is high. For capillary and small veins, hematocyte value is very low because rate of flow of blood is different in different vessels. So depending on the flow rate of flow of blood, you can see the hematocrit value. So that is what they explained here. If the value is very less, the blood flow in the veins will be very less. If the value is high, the blood flow in the veins will be high. So that veins can be identified depending on the flow of blood. If the larger veins andre, it may be aorta or veins, and smaller veins such as capillary veins, it can be identified. So this is with respect to effect of viscosity on nature of blood flow through different vessels. Next detection of intra-disulfide bonds in protein. So proteins ಅಲ್ಲಿ how you are going to identify the intra-disulfide bonds. So how it may be formed andre. So intra-disulfide bonds in the proteins can be detected through various methods, which includes mass spectroscopy and also X-ray crystallography. So also with respect to computational methods, also it can be identified. Computational method and it's nothing but by computer-based work. So some theoretical works are there depending on that computational method. By using some of the softwares, they are going to identify the structure. So analyze how it in the presence of disulfide bonds in the protein based on the amino acid sequencing. How the disulfide bonds are formed andre it's nothing but proteins ಹಿಂಗೆ ಫಾರ್ಮ್ ಆಗಿರುತ್ತೆ. It's nothing but the sequencing of amino acids. One amino acid ಇಂದ ಇನ್ನೊಂದು amino acid linkage ಆಗ್ಬೇಕಾದ್ರೆ there will be formation of disulfide bonds will be formed. So that bonds formation can be identified. So that is with respect to detection of intra-disulfide bonds in the protein. So disulfide bonds andre it's nothing but S dash S bond formation with another protein group or with another substrate. So sulfur bond formation you can see. Next is gel electrophoresis. Gel electrophoresis it is a another electrophoresis you should be knowing what is electrophoresis andre. Then only you can be knowing electrophoresis. It is the migration. Electrophoresis andre it is the migration and suppression of charged particles under the influence of electric field. So it can be separation. So it is also a method. Electrophoresis andre it is a method of separation of charged particles under influence of electric field. So here we are going to study about the gel electrophoresis method. In gel electrophoresis, it is a method used to separate and analyze biomolecules such as DNA, RNA, proteins based on their size and charges. So gel electrophoresis is basically used to identify and separate the DNA, RNA, protein based and their molecules can be separated. So why you need to separate under to identify the structure and mass of that protein or a molecule. So the process involving this place of biomolecule in gel matrix applying the electric field. So you are going to take one gel matrix and that you are going to apply the electric field. So whatever the molecule present, you are going to plot on that gel matrix plot. So the biomolecules will migrate through gel towards the electrode on opposite charges. The biomolecules will migrate andre when the charge is on, the biomolecules will move towards the higher or lower surface of the gel depending on their molecular weight structure. And so if the small molecules will migrate faster andaga, the there will be larger further than larger molecules. So large molecules, so small molecules, so it gets separated. So you can see the gel electrophoresis method. So here smaller molecules will migrate faster and farther than larger molecule. So small molecules will migrate faster. You can see this blot. This blot will be kept inside this tub and there will be electric field passing toward that blot. So there are different types of gel electrophoresis such as agarose gel electrophoresis for DNA and RNA polymerization and DNA and RNA separation and page that is polyacrylamide gel electrophoresis. So one more is polyacrylamide gel electrophoresis for protein separation. So the protein separation is based on the size and charge. So protein can be separated based on their molecular weight, size and charge, also with respect to molecular weight. So in gel electrophoresis, there are two different methods. That is agarose gel electrophoresis for the separation of DNA and RNA. DNA and RNA separate ಮಾಡೋದಕ್ಕೆ agarose gel electrophoresis method we are going to use. And one more that is polyacrylamide gel electrophoresis that is generally used for protein separation. So the protein separation is nothing but the separation of amino acids inside the protein. So this protein is based on the some this protein separation mainly how it is separated and right based on the size and charges, also with respect to their molecular weight. So coming to the gel electrophoresis, it is widely used in molecular biology or biochemistry for a variety of applications such as DNA sequencing, mutation detection, and protein analysis. So recently I've seen with COVID-19 where mutation of DNA and RNA sequencing they have created the virus. So you can be separated and changing of combination of DNA and RNA combinations can be done using this gel electrophoresis method. So it can be separated. Also it can be mutation. It can be joined with another sequencing. The sequencing can be changed here. So depending on this, the gel electrophoresis is used. So here the selection of appropriate gel. So the methods how we are going to conduct this experiment andre. So first preparation of gel electrophoresis. So in that selection of appropriate gel matrix we should be selected. That is agrogel that is used for DNA or RNA suppression. Polyacrylamide gel that is basically used for protein suppressions. So two different suppression DNA RNA we are going to use agarose gel, and protein suppression we are going to use polyacrylamide gel. So same method we are going to use different gels for the separation. So the preparation of gels, how we are going to prepare those gels andre it dissolved in agarose powder is a buffer solution heating until the completely dissolved, and further it is poured into gel casting tray and allowed to solidify. So gel casting tray we are going to dissolve and pour it. It will get solidified. So further in the same way with polyacryl acryl amide gel no same method alle we are going to prepare. We are going to dissolve and further eating until it completely dissolve. So completely dissolve ಮಾಡಿ you are going to pour into a gel casting tray and loading the sample. The sample is separated. So whatever the samples needs to be separated, those are denoted as sample here. Samples under loading the samples under the samples which need to be separated, such as DNA, RNA, sequencing or proteins we are taking here and mix it with loading buffer, uh, which contains tracking dye. For dye, we are going to use different types of dyes to identify the separation. ಈಗ lower bands, longer bands, shorter bands ಅಂತ ಬರುತ್ತಲ್ಲ ಇಲ್ಲಿ. So depending on the colors, we are going to use different dyes to be separated, and the samples to be separated is mixed with loading buffers, which contains a tracking dye to allow visualization of migration. So migration from lower bands to shorter bands ಇಲ್ಲ longer to shorter range ಗೆ shift ಆಗೋದನ್ನ we can identify easily. So we are going to use the dye with the samples ನ inject ಮಾಡ್ಬೇಕಾದ್ರೆ. So depending on the samples through gel, the sample is then loaded onto gel using micropipette. Using micropipette, we are going to ಇಲ್ಲಿದೆ ನೋಡಿ how we are going to make ಅಂದ್ರೆ see this one is with respect to gel solution, gel and slots containing DNA separation. ಈ starting ಇದೆಯಲ್ಲ, so here we are going to pin ಅಂದ್ರೆ one pinch we are going to on that gel tray ಮೇಲೆ we are going to insert ಅಂದ್ರೆ using micropipette. So when you start or insert with the current, they start moving and get separated. ಈಗ ಪೇಪರ್ ಕ್ರೋಮಟೋಗ್ರಫಿ ನೋಡಿದಿರ ಅಲ್ವಾ, ಇಟ್ ಮೂವ್ಸ್ ಅಪ್ವರ್ಡ್ಸ್ ವೈಲ್ ಯು ಇನ್ಸೈಡ್ ದ ಸೊಲ್ಯೂಷನ್. ಸೇಮ್ ವೇ ಇಟ್ ಗೆಟ್ ಸಪರೇಟೆಡ್. ದ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಹೆಂಗ್ ಸಪರೇಟ್ ಮಾಡ್ತೀರಾ ಪೇಪರ್ ಕ್ರೋಮಟೋಗ್ರಫಿಲಿ, ಡಿಪೆಂಡಿಂಗ್ ಯು ಆರ್ ಗೋಯಿಂಗ್ ಟು ಪ್ಲಾಟ್ ಆನ್ ದ ಎಂಡ್ ಅಲ್ಲ ಒಂದ್ ಸೈಡ್ ಅಲ್ಲಿ. ಸೊ ಯು ವಿಲ್ ಬಿ ರನ್ನಿಂಗ್ ವಿತ್ ದ ಸಮ್ ಆಫ್ ದ ಸೊಲ್ಯೂಷನ್ಸ್, ಪೋಲಾರ್ ಅಂಡ್ ನಾನ್ ಪೋಲಾರ್ ಸೊಲ್ಯೂಷನ್ಸ್ ನ ಇನ್ಸರ್ಟ್ ಮಾಡಿದಾಗ you are going to get different bands. So the all the molecules will be getting separated. Here using the gel electrophoresis method, we are going to separate the RNA and DNA. Also the protein sequencing can be separated. So these are the separated molecules that can be seen in the tray here. So after electrophoresis is complete, the gel is removed from the chamber and strained with reagent to visualize the separated biomolecules. Separated biomolecules now we are going to see Andre. These are each dots what you are seeing here. It is showing separated different biomolecules. So that identify ಮಾಡೋದಕ್ಕೆ we are going to dip inside the ethidium bromide solution. It is commonly used for DNA or RNA separation. So that solution ಒಳಗೆ insert ಮಾಡಿದಾಗ we are going to get the different band colors. So you can identify different DNA and RNA present inside that sample. So samely it can be used for protein separation also. So the analysis of results we are going to analyze this under the separated biomolecules can be visualized under UV light or UV chamber by SFI, and the position on the gel can be used to determine the size and identify the specific molecules. So the position of the gel can be used to determine their size and or identify the specific molecules. And DNA sequencing, the gel electrophoresis ಆಯ್ತು. So next is DNA sequencing. DNA sequencing, it is the process of determining the order of nucleotide in a DNA molecule. So to determine the order of nucleotide means DNA the order identified, that is known as DNA sequencing. So it is widely used to know the Sanger sequencing or deoxy chain termination method. So the Sanger sequencing procedure has several steps. So DNA sequencing has several steps on that. So DNA sequencing here first to DNA preparation. The DNA sample is first purified, and whatever the how you are going to purify the DNA andre how you are going to extract the DNA. You are going to extract from the ಎಲ್ಲಿಂದ air blood ಇದರಿಂದ ತಾನೆ. So first you are going to extract DNA and purify that sample, and further the fragmented then DNA fragments are copied. So in this process, first purification of DNA sample and template preparation, DNA fragments are then used to template DNA synthesis using DNA polymerization or modified nucleotide. DNA polymerization is nothing but synthesizing of nucleotides. Nucleotide synthesis na DNA polymerization another. So chain termination. So we are going to use dideoxynucleotide are added to reaction mixture which will terminate the DNA synthesis at specific position. So whatever the sequence created, DNA polymerization in the ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಚೈನ್ ಫಾರ್ಮ್ ಆಗಿರತ್ತಲ್ಲ ಅದು ಸೀಕ್ವೆನ್ಸ್ ನ ಅಂದ್ರೆ ಆ ಚೈನ್ ನ ಕಟ್ ಮಾಡೋದಕ್ಕೆ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಯೂಸ್ ಡಿಆಕ್ಸಿ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್. ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಯೂಸ್ ವಿಲ್ ಟರ್ಮಿನೇಟ್ ದ DNA sequencing. So that is what DNA sequencing ಅಂದ್ರೆ. ಈಗ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ DNA will be of a circular or linear. So you need to identify or sequencing. The sequence should be breaked ಅಂದ್ರೆ ಚೈನ್ ನ ಕಟ್ ಮಾಡ್ತೀರಾ. ಸೊ ದಟ್ ಚೈನ್ ಕ್ಯಾನ್ ಬಿ sequencing can be done ಯಾವ್ದ್ರಿಂದ by using dideoxynucleotide are added to reaction mixture which will terminate the DNA synthesis at specific position. So DNA synthesis ಆಗೋದನ್ನ can be stopped. So DNA sequencing can be separation. So separation of DNA fragment resulting the fragments are then separated by size using gel electrophoresis. So whatever the DNA synthesis or sequencing can be done means it is nothing but adding of nucleotide. Nucleotide na add ಮಾಡ್ತಾ ಹೋಗ್ತಾ ಇರ್ತೀವಲ್ಲ, that is what DNA polymerization ಅನ್ನೋದು or sequencing. So that can be stopped. ಅದನ್ನ ಫಸ್ಟ್ ರಿಯಾಕ್ಷನ್ ನ ಸ್ಟಾಪ್ ಮಾಡೋದಕ್ಕೆ we are going to use dideoxynucleotide can be added to the reaction mixture which will terminate the DNA synthesis at specific position. So addition of nucleotide ಇದಕ್ಕೆ ಮತ್ತೆ add ಆಗ್ತಾ ಹೋಗುತ್ತೆ nucleotide that can be stopped. Further, how you are going to separate this? ಈಗ synthesis ಆಗನ್ನ again you need to separate identify ಮಾಡಬೇಕು ಅಂದಾಗ then you are going to go back to gel electrophoresis method. So in the previous method by DNA RNA can be separated here. This by this method can be separated. So DNA gel electrophoresis method is used. So detection. So after that, how we are going to detect this bands andre the separated DNA fragments are then visualized using the UV chamber. So it can be a sequence determined by reading the order of nucleotides from bottom of the gel to the top. So from bottom to top, the order of the nucleotide can be identified. And other method here is these methods are generally used for large-scale DNA sequencing projects. So such DNA sequencing, transcription sequencing, and metagenomics. So this is for DNA sequencing. Next is DNA fingerprints. DNA fingerprinting, also known as DNA type of genetic fingerprinting. It is a technique used to identify the individual based on their unique DNA profile. So DNA fingerprints it can be identified on based individual based on their unique DNA profile. By analyzing their variations, it is possible to create unique DNA profile for each individuals. So each individuals it can be possible to create different DNA profile. The process of DNA fingerprinting is seen here first extraction, suppression, restriction of the how we are going to extract DNA ಅಂದ್ರೆ it is extracted from blood ಅಥವಾ saliva ಅಥವಾ air or other tissues. The DNA is extracted, then the sequencing has been done, then further it is separated the DNA. Each step ಇಲ್ಲಿದೆ ನೋಡಿ how the step process is taking place ಅನ್ನೋದು. So extraction, sequencing, separation, and DNA sampling. Further, electrophoresis, gel electrophoresis, where it can be separated. Long DNA transfer fragments and short DNA fragments can be separated. It's nothing but DNA, the nucleotides, alla. So nucleotides can be separated and further transferred to membranes and incubation in the label protein. You can see the pattern. So this is the patterns of separation of the DNA. So first in the first step, we are going to extract from blood, saliva, air, and PCR amplification. PCR here is polymerase chain reaction. So DNA is then amplified using technique called polymerase chain reaction, which makes many copies of HTR regions. So and further suppression and detection. So DNA is amplified first using technique called polymerase chain reaction (PCR technique), and further amplified DNA is then separated by size using gel electrophoresis. So each step they are given in the explanation and the separated by gel electrophoresis and identify using UV chamber. and separated DNA bands and the profile creation. The pattern of bands in the gel is used to create a DNA profile for individual. So DNA profiles can be created using the gel electrophoresis and comparison. The DNA profile is then compared to profile from other individuals. So DNA fingerprints. So how you are going to match each other and you are going to create a separate profile and match each other whether the DNA is matching or not another. So depending on this, you are going to identify the by each other's DNA profile. So DNA fingerprint is widely used for forensic science and paternity testing and other areas where identification of individuals is important. For his DNA can be fingerprinting can be used. So in this method, for the identification, forensic science, paternity testing, and also other areas where identification of individuals can be done using DNA fingerprint. Next to conformation of nucleic acid. So nucleic acid such as DNA, RNA are long linear polymers made up of nucleotides. Nucleotides are nothing but nucleic acids such as DNA and RNA. So some of the nucleic acid contains sugar, phosphate group, and ultra nitrogenous base. So depending on the conformation of nucleic acid refers to three-dimensional structures. So here we are going to conformation of nucleic acid. Conformation of nucleic acid can be on the three-dimensional structure or shape. This is most well-known conformation DNA in the double helix. So two complementary strands for DNA and opposite direction are held together by hydrogen bond formation between the base adenosine triphosphate AT and CG for the Rux ladder like structure. So in the RNA, in the other end, adopts different conformation based in on its function. So the conformation of nucleic acids plays very important role in the function. Why this conformation is important, Andre? It is important to play an important role in the functioning, such as determining the accessibility of the base to enzymes, determining the rate of transcription and translation, and aiding the regulations of gene expressions. So, determining the gene expressions, the conformation of nucleic acid is much important. DNA and RNA. Next is the protein charges. So proteins are nothing but the enzymes. So proteins are mainly building blocks of amino acids. So sequencing of long amino acids are known as proteins. Proteins ಏನಿಲ್ಲ. It's a combination of 21 amino acid different combinations ಅಲ್ಲಿ ಲಿಂಕ್ ಆಗಿರುತ್ತಲ್ಲ. So that amino acids links bulk amino acids are known as protein. So proteins are complex biomolecules made up of amino acids where which are joined together. Formation of peptide bond will be formed. So the sequencing of amino acids are known as proteins. Simple way andre proteins are building blocks of amino acids. So the properties of proteins are determined by its amino acid sequencing. So different sequencing irutte. Each amino acids linked with another amino acids. Different amino acids link adaga different proteins are formed. So which in turn is determined by genetic coding. So amino acids can be classified into several groups based on their chemical properties. So amino acids considered to be acidic if it is having carbonyl group. Some of the amino acids which are present with the carbonyl amino group and some amino group are present. Sulfur groups are present. Hydrogen. So depending on the groups present, it can be separated. So carbonyl group present idre, it is more acidic in nature, and amino group are present andre it is more basic in nature. So the overall charges of protein can be positive, negative, or neutral. So in this method, we are going to identify the charge on the proteins. Protein the charge how we are going to identify and depending on the groups present inside the amino acid. Amino acids ಅಲ್ಲಿ carbonyl group idre it will be more acidic, and amino group NH2 is present andre it will be more basic. So on that we are going to initiate. Acidic idre ಏನಾಗುತ್ತೆ, it will be of negative charge. Basic andre it will be of more positive charges. So first we are going to initiate ಐಡೆಂಟಿಫೈ ಮಾಡ್ತೀವಿ. ಈಗ ಅಮಿನೋ ಆಸಿಡ್ಸ್ ನ ಪ್ರೋಟೀನ್ಸ್ ಅಲ್ಲಿ ಅಮಿನೋ ಆಸಿಡ್ಸ್ ಪ್ರೆಸೆಂಟ್ ಇರುತ್ತಲ್ಲ, ಅಮಿನೋ ಆಸಿಡ್ ನ ಫಸ್ಟ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಐಡೆಂಟಿಫೈ ವೆದರ್ ಇಟ್ ಈಸ್ ಹ್ಯಾವಿಂಗ್ ವೆದರ್ ಇಟ್ ಈಸ್ ಅಸಿಡಿಕ್ ಆರ್ basic, and depending on that nature we are going to say whether it is in having positive charge or negative or neutral charge. Amino acids ಏನಾರು ನೆಗೆಟಿವ್ ಅಲ್ಲಿ ಇದ್ರೆ it is having more carbonyl group ಅಂದ್ರೆ it will be in the negative side. So negative ಅಂದಾಗ and so the proteins which are having more acidic amino acids, then it will be having negative charges. Amino acids which are having negative, the amino acids which are in acidic, amino acids which are in acidic andre which are present which are having carbonyl group. The amino acids which are having amino group which are in basic, so acidic andre it will be having negative charge. So acidic andre it will be having negative charge. Amino that is basic group. So that group will be the proteins which are having more basic amino acids that will be in the positive charge. So depending on the acidic or basic, the proteins charges can be identified. Protein which is having negative or positive charge depending on the again groups present in that. So whether it is having negative or positive. So it's important ಏನಕ್ಕೆ why to identify the charges present inside the protein is to identify andre the charges of protein can affect by factors and pH. So it can be the protein though charges it can be affected. Also the pH, depending on the pH also with respect to temperature, the presence of other molecules such as ions, and this change in charge can affect the protein's activity. So this is the end of the unit.